how does a cluster of completely identical stem cells somehow know how to build a human spine and a spinal cord at the exact same time in perfect synchrony?
Well, it's arguably the most complex construction project in the known universe.
I mean, you're sitting there right now listening to this, and your nervous system is perfectly threaded through the bony armor of your vertebrae.
Right, and to get to that point, millions of cells had to coordinate this microscopic ballet in the dark, completely without a choreographer.
Which is wild. And it brings us to our mission for today's deep dive.
Right.
We are looking at a truly groundbreaking paper published in Nature Cell Biology in 2025.
Yeah, by Makwana and colleagues at the Francis Crick Institute.
Right, and they essentially managed to crack open that black box, right?
Yeah.
They figured out how to recreate the specific human tissues that build the trunk of the body.
They did. They created what they call human trunk-like structures, or hTLS for short.
And the why here is really crucial for context, because we're zooming in on a very specific, highly critical window of your own creation, which is Carnegie stage 13 to 14.
Which is roughly day 28 to day 35 after fertilization.
Right. And at this stage, tissues aren't just growing in isolation, they're engaging in what we call co-development. Okay, let's unpack this. Co-development. What does that actually look like on a cellular level?
Well, it means neighboring tissues are essentially talking to each other, like they are trading chemical blueprints back and forth to ensure they grow properly.
Like checking each other's work.
Exactly. If one tissue gets lost, the other one is supposed to, guide it back on track.
So if you were a stem cell, just a blank slate, you have to figure out what to become. In the trunk of the body, there are basically two main players we need to care about today.
Right. First, you have the neural tube, which is the tissue that will eventually fold and zip up to become your spinal cord and your brain.
And second, the somites.
Yeah, the somites. These are these little paired blocks of tissue that run down either side of the neural tube.
And they eventually become like your vertebrae, your ribs, and your skeletal muscle.
Right. That's right. And historically, if scientists wanted to understand how the neural tube and the somites talk to each other, they had to rely on classical embryology.
Which means what exactly?
It meant taking animal embryos, like chicks or mice, and literally cutting and pasting tissues. They would surgically move a piece of a mouse's neural tube to a different spot to see what would happen to the surrounding somites.
Oh, wow. But doing that in human tissue is a whole different ballgame.
Exactly. Doing this in human embryos is both technically unfeasible and ethically constrained. I mean, for decades, researchers operated under the famous 14-day rule.
Right, which strictly limited the culture of human embryos in the lab to just 2 weeks.
Exactly.
But wait, the developmental window we're talking about for the spine is around day 30, right? So we were just completely locked out of seeing it happen. We had a missing chapter in our own biological history.
We did, and that is exactly why they had to build these stem cell-based embryo models.
Okay, let me push back here for a second, because whenever people hear embryo models, they panic. Are these scientists actually growing a tiny human in a petri dish?
No.
Like, does this bypass those ethical rules entirely, or are we entering a scary new gray area?
It's a really critical distinction to make, and the answer is a firm no. These are purely in vitro models.
Okay, good to know.
Yeah. The researchers take pluripotent stem cells, which are cells that have the potential to become almost anything in the body, and they coax them into highly organized 3D clusters.
But they only mimic very specific tissues, right?
Exactly. They absolutely do not form a complete embryo. There is no brain, there is no beating heart, and crucially, there are no extra embryonic tissues.
Like a placenta.
Right. Which are strictly required to form a viable pregnancy. It's a localized, completely isolated simulation of just the trunk.
Okay, that makes sense. So to build that specific trunk, how did they know which cells to use?
Well, they used a brilliant tool called single-cell RNA sequencing. Think of it as reading the specific instruction manual each individual cell is holding at any given second.
Oh, so by looking at the RNA, they can see exactly which genes are turned on and which are turned off.
Precisely. And through this, they found the mother lode, which are neuromesodermal progenitors. Let's just call them NMPs.
NMPs.
Got it.
What makes them so special?
They are the ultimate dual-purpose starting blocks. If you look closely at these NMP cells, they express two very specific genes at the exact same time.
Okay, what are they?
SOX2, which is essentially a chemical tag that says, I have the ability to become neural tissue, and TBXT, a tag that says, I have the ability to become mesoderm, which makes the somites.
Oh, wow. So they are literally sitting on the fence. They are the shared ancestral pool feeding both structures.
Yes. And what's fascinating here is that by identifying that shared pool, scientists realized they didn't need to manually build a neural tube and somites separately and then stick them together like Frankenstein's monster.
They just needed to give the NMPs the right environment, and the cells would sort it out themselves.
Exactly.
Okay, so let's hit our first checkpoint here. I want to do a rapid-fire bulleted summary of what we just covered right around the 5-minute mark of our deep dive.
Good work.
Okay, bullet 1. Tissues need to talk to each other to grow, which is co-development. Bullet 2. We can't study 28-day human embryos easily, so scientists built a stem cell model. And bullet 3. NMP cells act as the dual-purpose starting blocks.
That's a perfect summary.
So here is a question for you listening right now. Think about the building blocks of your own body. If you were a scientist, how would you even begin to coax stem cells into these complex shapes?
It's a great question because finding the starting block is really only half the battle. To actually coax those blank stem cells into these specific shapes, you need a very precise biochemical recipe.
Right, because you have this cluster of stem cells in a dish, but they are just like a disorganized pile. How on earth does a scientist actually force them to build a spine?
Well, they have to manipulate the cell's internal signaling pathways. And this team absolutely nailed the recipe.
Okay, wait on me.
You start with those human pluripotent stem cells. Then you introduce an activator for a signaling pathway called WNT.
WNT, W-N-T, right?
Right. The specific chemical they use is called CHIR99021, but let's just call it Chi.
Okay, so we add Chi to activate the WNT pathway.
Right. Then you add an inhibitor for a different pathway called nodal. They use a chemical called SB431542, or SB43.
So you have Chi activating WNT and SB43 blocking nodal.
Got it. Next, you embed this entire cellular cluster into a gel made of 10% matrigel.
Matrigel, what does that do?
It provides a 3D physical scaffolding. Cells need physical tension to organize.
Oh, like they need something to push off of.
Exactly. Without matrigel acting as an extracellular matrix, literally giving the cells something to grab onto and pull against, they just form a disorganized blob. They need to know up from down, and physical tension provides that map.
That is wild. Okay, so what's the last ingredient?
To top off the recipe, you pulse the whole environment with retinoic acid.
Okay, so we've got Chi activating, SB43 inhibiting, matrigel for scaffolding, and retinoic acid.
Yeah.
I want to go back to that WNT activator, the Chi chemical.
Sure.
Because based on the notes, I love looking at this like an audio mixing board. Does that Chi chemical basically act like a volume knob?
That is a perfect analogy, yes.
Yeah.
The researchers found that if you crank the WNT signaling up really high, meaning you use a high concentration of Chi, the stem cells essentially get deafened to any other signals.
So they just get overwhelmed by the noise.
Exactly. The WNT signal is so loud that every single cell goes down the mesoderm pathway. You end up with a cluster made entirely of somites. No neural tube whatsoever.
Wait, really? But what if you turn that WNT volume down?
Well, if you lower it to a very specific, quiet level, you allow for what we call modularity.
Modularity.
Yeah. The lower chemical noise lets the cells differentiate into different things. Suddenly, a beautiful neural tube forms right alongside the somites in the exact same dish.
Okay, so why does that matter so much? I mean, it sounds like a neat trick, but what's the actual scientific value?
Think about it from an experimental design perspective.
Right.
Because they have a volume dial, they can essentially play biological Lego.
Oh, they can create a tissue model that is just somites and another one right next to it that is somites plus a neural tube.
Exactly. They can run A/B testing on human tissue development.
That is so clever.
Right. If you want to know if the neural tube is sending a crucial signal to the somites, you just turn the volume up on one dish to erase the neural tube and see if the somites still develop normally.
And if something goes wrong in the somites, only when the neural tube is missing. Boom, you've just proved the neural tube was providing a vital instruction.
Exactly.
That is genius. Okay, checkpoint 2, let's do our mini recap. Bullet 1, the recipe requires WNT activation, nodal inhibition, and matrigel. Bullet 2, modularity allows scientists to play biological Lego, adding or removing the neural tube by adjusting chemical concentrations. So, listener, if you had a biological volume dial for your own cells, What other tissues do you think you could coax into existence just by changing the chemical noise? It really makes you appreciate the morphological magic happening inside us.
It really does. And the word morphological is key here because the physical transformation these cells undergo is just stunning.
Yeah, I was looking at the images from the paper.
When you look at these hTLS models under a scanning electron microscope, you aren't just looking at a flat layer of cells, you are looking at architecture.
It is wild. If you picture looking at the very bottom, the tailbud region of this growing cluster under the microscope, the cells there are really loosely packed. They look chaotic, almost like a pile of wet sand.
Yes, very disorganized.
But then, as you move your eyes up the structure into what is called the presomatic mesoderm, or PSM, the cells start packing closer together. It's like medium compaction. And then at the very top, it is absolute structural perfection. You see tightly packed, beautiful epithelial blocks, which are the somites, flanking a perfectly smooth, contiguous pipe, which is the neural tube.
That transition from a loose pile of wet sand to a tightly bound epithelial tube is profound, particularly when we talk about the neural tube.
How so?
Well, I want to dive into the mechanism there because in mammalian development, the neural tube actually forms in two completely different ways depending on where it is in the body.
Wait, really? It doesn't all form the same way?
No, it doesn't. The upper part of your spine and your brain forms through something called primary neurulation.
Primary neurulation, okay.
This is where a flat sheet of cells physically rolls up and zips closed down the middle. When primary neurulation fails, that zipper gets stuck and you get severe neural tube defects, like spina bifida.
Oh, wow. Okay, so that's the upper spine. What about the lower trunk, which is what this hTLS is modeling?
The lower trunk uses secondary neurulation. There is no flat sheet folding up. Instead, the cells condense into a solid, dense rod of tissue.
But a rod isn't a tube. How does it hollow out?
Little pockets of fluid start to appear inside the solid rod. The researchers tracked a protein called Z01, which naturally coats the inside of these tiny fluid pockets.
So, if I'm picturing this right, imagine you're sitting in your car while it's raining, watching water droplets on the windshield. When two droplets get close enough in touch, they suddenly merge into one bigger droplet. Is that what's happening inside this solid rod of cells?
That is a perfect visual. The cells form separate little fluid-filled cysts, and over time, as those cysts grow, they progressively merge together, just like the raindrops.
That's incredible.
Yeah, eventually they hollow out the entire solid rod until it becomes one long, contiguous pipe. Witnessing this secondary neurulation happen in real time in a human model is a massive leap forward for biology.
Because we've never been able to observe the mechanics of it so clearly.
Exactly.
And while that pipe is hollowing out, the somites next door are doing their own highly coordinated dance. But here's what I don't get.
What's that?
If they start as a continuous line of tissue, how do the somites know exactly when to pinch off into perfect little individual blocks? It clearly isn't random.
Far from it. It's governed by a highly precise biological clock called the segmentation clock.
A clock, like an actual timer inside the cell.
Yes, driven by gene expression. The researchers used a fluorescent reporter line, basically attaching a glowing tag to a specific gene called the hES7 gene, so they could watch it turn on and off under the microscope.
How does a gene act as a clock?
It's a feedback loop. The gene turns on, it creates a specific protein. Once that protein reaches a certain high level in the cell, it actually triggers its own gene to shut off.
Oh, I see.
Then, as the protein degrades and disappears, the gene turns back on again. On, off, on, off. That's a clock.
That makes sense.
And what they saw was a literal wave of this gene expression sweeping through the tissue, moving from the posterior tail up to the front. And it happened on a remarkably strict schedule.
They measured the exact timing, didn't they?
Yes.
A new wave sweeps through every 298 minutes. That is roughly every 5 hours.
Yes.
It is literally a biological metronome ticking inside the cells. Every 5 hours, tick, a new block of tissue pinches off, tick, another vertebra is born.
And if we connect this to the bigger picture, it proves that the intrinsic rhythm of human development is encoded deep within the DNA of these progenitor cells.
They don't need external cues.
No, they don't need a mother's heartbeat. They don't need external hormonal cues to keep time. The 5-hour clock is built into the genetic circuitry itself.
Okay, checkpoint 3. Let's synthesize this.
Bullet 1.
The tissue physically changes from loose cells to tightly packed epithelial tubes.
Right.
Bullet 2. The neural tube forms via secondary neurulation, which is cysts merging into a pipe like water droplets.
All right.
And bullet 3, a genetic clock picks every 5 hours to generate new somite blocks.
Perfect.
So, listener, Imagine a clock ticking inside your cells right now. How does it change your perspective of your own body to know you were built to a 5-hour rhythm?
It really brings a profound sense of order to something that feels so mysterious. And that order relies entirely on communication between the structures.
Right, because here is where it gets really interesting. We've got our 5-hour clock ticking, our somites forming, and our neural tube hollowing out. But they are sitting right next to each other in this dish. How are they actually talking?
Well, to understand the conversation, we first have to notice who is missing from the room.
Who is missing?
In a natural, fully formed embryo, there is a specialized rod of tissue right beneath the neural tube called the notochord.
Okay, the notochord.
The notochord acts like a localized beacon. It constantly shouts out a chemical signal to tell the bottom of the neural tube, hey, you are the bottom.
Oh, like giving it directions.
Yes, this process is called ventralization, giving tissue a belly or bottom identity. But our hTLS model, because it is just a partial simulation, doesn't have a notochord.
Ah, so because the notochord isn't there acting as an anchor, does the neural tube get confused about which way is up?
Exactly. It defaults to what we call a dorsal identity. It thinks the whole thing is the top or the back of the spinal cord. It lacks a bottom.
That's fascinating.
But the scientists wanted to see if the walkie-talkie receivers on these cells still worked. So they manually added the missing chemical signal that the notochord usually produces. It's a very famous protein called sonic hedgehog, or SHH.
Wait, scientists actually named a crucial developmental protein after Sonic the Hedgehog.
They did. Biologists have a sense of humor, too.
I love that so much. Okay, so they drop Sonic Hedgehog into the dish. What happens?
The tissue responded beautifully. When they added SHH, it effectively ventralized the tissue. It gave a bottom or belly identity to both the neural tube and the adjacent somites.
So the receivers worked.
Yes. Furthermore, it happened in a dose-dependent manner, meaning the more SHH they added, the stronger the ventral identity became. It proved the receiver on the walkie-talkie was fully functional, even outside of a real embryo.
But they also found a conversation happening completely spontaneously, right? Without the scientists adding anything.
Yes, they did.
And looking at the notes, this reciprocal feedback loop seems like the absolute crown jewel of the paper. Let's walk through this loop step by step because the mechanics is just elegant.
Let's do it.
Step 1. The neural tube sends a cue out into the void, specifically targeting the inner side of the somites right next door.
Right, and step 2. The somites receive that cue across the gap, and they respond by switching on a specific gene called ALDH1A2. You can think of ALDH1A2 as a specialized manufacturing plant.
Okay, step 3. That ALDH1A2 factory starts pumping out retinoic acid, or RA, and this retinoic acid physically diffuses out of the somite, travels back across the gap, and hits the neural tube.
And here is the payoff, step 4. When the retinoic acid hits the neural tube, it binds to receptors that trigger the expression of a critical neural gene called PX6.
PX6.
Yeah, and PX6 is the master switch that kicks off neurogenesis, the actual creation of mature neurons.
Wow. So it's a perfect loop. The neural tube essentially pokes the somite and says, hey, I need something. And the somite turns on its factory and spits out retinoic acid. And then the retinoic acid travels back to the neural tube and says, message received, you have permission to start making neurons. They are completely codependent.
They are. And this raises a massive paradigm-shifting question for developmental biology. If we connect this to the bigger picture, for decades, we assumed you needed the massive complex architecture of a whole, intact embryo to study how human organs learn to function together.
Right, because everything is so interconnected.
Exactly. We thought the signaling required the whole system. But this spontaneous, self-organized signaling loop proves that is false.
Because they did it in an isolated dish.
Precisely. The local tissue neighborhoods have enough inherent intelligence to set up their own communication networks as long as they're in the right proximity.
Okay, checkpoint 4. Let's lock in this final piece of the puzzle. Bullet 1. Without a notochord, the tissue is dorsally biased but perfectly responds to SHH. Bullet 2. The neural tube and somites form a feedback loop using ALDH1A2 and retinoic acid. In bullet 3, this retinoic acid is required to trigger neurogenesis, making the neurons.
That's right.
So listener, think about the feedback loops in your own life. How often do you require a signal, a word of validation, or a cue from someone else before you can take your next step forward, just like these developing tissues?
It's a great analogy.
So what does this all mean? beyond just being a fascinating biological parlor trick, the real-world clinical application here is immense, isn't it?
It really is. Remember, early on, we mentioned these models are built from human-induced pluripotent stem cells, iPS cells.
Right, the stem cells that can become almost anything.
Exactly. That means we could, in theory, take a simple skin swab from a living patient who suffers from a rare neural tube defect.
Oh my goodness. We can revert those skin cells back to stem cells and then grow a custom hTLS model of that specific patient's embryonic spine.
Yes. We can literally wind the clock back. We can watch their personal day 30 of development under a microscope and find the exact hour, the exact chemical walkie-talkie signal that failed.
That is just staggering.
It moves embryonic research from a generalized, abstract study of human development into the realm of highly personalized medicine. We can pinpoint whether their specific failure was a broken ALDH1A2 factory in the somite, or maybe a deaf PAX6 receiver in the neural tube.
It brings us right back to that initial image, millions of cells pulling off a synchronized ballet in the dark. Only now we finally have a window to watch the performance.
And maybe even fix the choreography when it goes wrong.
Exactly. Thank you so much for joining us on this deep dive. Keep questioning the incredible microscopic choreography happening inside you right now.
And I'll leave you with this final thought. If we can fully map out the chemical walkie-talkies between the embryonic spine and the spinal cord, could we eventually use these specific signaling loops, like the retinoic acid and PAX6 pathway, to treat adults? Imagine regenerating a severed spinal cord by tricking the damaged tissue into thinking it's day 30 all over again, coaxing it to co-develop anew. The blueprint is already inside us. We just have to learn how to read it.